Good morning, everyone, and thank you very much for your coming and in person, and also someone online. And today is a great pleasure for me and to introduce our special speaker, Dr. Mirek Styblo. And we know each other not that long, and but I feel it's like we are a very good old friend. And because he's one of a few can very correctly pronounce my last name, Luk Tai, and not that many people. And hopefully, and I can pronounce correctly too for your name today. Yeah, and Dr. Styblo and received his bachelor and Ph.D. in 1980 and in 1988, and later in the 1992, both and in the human chemistry and chemistry and for major. And later in 1992, and he moved to the United States and got his postdoc training in the US EPA and until in 1995. And currently, Dr. Styblo and as a professor and work in the how to pronounce that, Dealing School of the Global Public Health and at the University of North Carolina and at Chapel Hill. And in that school, and they have one unique program that called the BS-PhMS. So that basically is a 5 years training, and you got a bachelor and a master, and for your and the public health and environmental health degrees. So he is the director for that program. And he also is a adjunct and professor in the UNC, and as that is which department? That is like a environmental science and engineer and department. And he is also is a head of the division head and the metabolic and health, and that is in UNC Institute of the Environmental Science Health Solution. And he also is an assistant director for Superfund at UNC, and that is his assistant director. And Dr. Styblo's long-term interest is mitotoxicology and nutrition biochemistry. And for that is the research, and he has continually got the support more than 20 years, and support from the NIH and specifically CIEHS and the EPA and also European commissions. And his primary interest is that and advice in fact of the inorganic and the arsenic, and for that he is as a PI for several population and the laboratory study project that is to define and the factors that affect the arsenic metabolism and arsenic the association with the metabolic disease, and also is a mechanism underlying how the arsenic and affect the disease. And he's also the head of the as the head of the Gilbert Institute or School. They have developed a very novel and technique to measure the spatial analysis of arsenic in the complex and biological material. And Dr. Styblo has served several panels for the US and the EPA and NIESH and NTP and several government agency as the reviewer, advisor panels members. And that is predominantly where evaluate the health and the risk and association with the environmental exposure arsenic. And Dr. Styblo also authorized more than 160 publications, and that is in the peer review journal and proceeding articles and also book chapters. And today, Dr. Styblo will bring us a very exciting topic, and that is a humanized mouse model for arsenic toxicology. Yeah, please, thank you.
All right, good morning, everyone. Thank you for the introduction, and thank you, Dr. States and Dr. Tsai, for inviting me to Louisville. This is my first time in Louisville, and I like it here, especially after last night lecture on Kentucky whiskey. Thank you, Dr. States. It was very inspirational. So I'll be happy to come back anytime you want me, assuming that there'll be another lecture. Okay, so I'm here to talk about arsenic though. So you heard Dr. Thai tell you that my history of research is mainly on arsenic, and that's the topic of my work. I was told that you are so much familiar about arsenic that I don't have to go in depth for introduction, so I have just a couple of slides to show you that arsenic is all around us. It's the 20th most common element in human Earth, so that means it's pretty hard to avoid exposure to arsenic at some point and some levels. It originates from mineral ores and from metal ores and minerals like arsenopyrite, from which it leaches to underground water, becomes part of surface water reservoirs, and ultimately is fed to any organisms on Earth, including humans, and these organisms are known to biotransform arsenic to a wide variety of organic arsenic compounds. Now, in addition to natural occurring arsenic, there is a lot of arsenic coming from human activities. Those of you who remember early 2000s, you may remember that flying ash from coal burning plants was a big issue. It's not flying anymore, it's being stored in sludges, but still a problem, especially during flooding. Herbicides and pesticides, some of them are made with arsenic, inorganic or methyl arsenic, so golf courses are probably most arsenic polluted areas in the United States. There was a wood preservative that contained copper chromate arsenate, which has been discontinued years ago, but those constructions, including some constructions in households and in playgrounds, are still there. They contain a lot of arsenic. Arsenic additives are used in poultry and swine production. Now, roxarson is not the best example because it was also discontinued several years ago, but there are at least 2 other feed additives that are still being used that contain arsenic. Arsenic is used as an antimicrobial agent in plastics, and as you probably all know, arsenic has been used historically, okay, with other metals like mercury for treatment of various disorders. Fuller's solution is one example, historical example, but a newer example is trisanox, which is arsenic trioxide, which is being used very effectively and successfully for treatment of acute promyelocytic leukemia. And having said so, I need to point out that the major source of arsenic exposure for humans is drinking water. Now, this map is pretty old and probably needs some updates, but it shows you some hotspots around the world. Everyone heard about West Bengal in India or Bangladesh, where estimated 40 million people have been exposed to arsenic through deep wells. There are hotspots in the US, although not all of them are shown here. There are some in North Carolina, which is all green on this map. So it's drinking water that affects hundreds of millions of people around the world, that drinking water contaminated with arsenic. And as you probably all know, arsenic exposure from drinking water has been associated with a variety of diseases. Arsenic has been characterized or defined as human cancerogen associated with skin, urinary bladder, lung, and liver cancers, and possibly with other cancers. But there are also non-cancerous diseases that have been reported in arsenic-exposed populations: black foot disease, peripheral vascular disease has been reported from Taiwan and then also from Bangladesh on a smaller scale. Cardiovascular disease has been associated with arsenic. Metabolic disease, specifically diabetes mellitus, more recently is linked to arsenic exposure. So arsenic is one of the environmental diabetogens, not obesogens necessarily, but diabetogens for sure. There are diseases of peripheral and central nervous system, and also some respiratory dysfunction. So arsenic is a multi-target toxin. It attacks cysteines around the body, and you can imagine what it does to a cell function, which relies on cysteines in proteins or low molecular weight cysteine compounds to function, actually. So it's a definitely multi-mode of action kind of toxin. Now, interesting part of arsenic toxicology is its metabolism. Inorganic arsenic, which comes from drinking water, is metabolized by addition of methyl groups from S-adenosyl methionine to form monomethyl arsenic species, dimethyl arsenic species, and in some animals or species, also trimethyl arsenic species. There have been two different mechanisms proposed: the classical oxidative methylation mechanism proposed by Challenger in mid-19th century. More lately, Japanese investigators suggested reductive methylation. The point is that either mechanism shows production of these trivalent monomethyl and dimethyl arsenic species, which we know today are extremely toxic. In fact, in some endpoints, they are more toxic than the inorganic arsenic itself. So this is interesting aspect of arsenic toxicology. Then, while we need to methylate arsenic to get rid of it, because methylation increases excretion, mainly through urine, we also produce these toxic intermediates, which most likely, almost certainly, contribute to adverse effects of arsenic exposure. Now, all this pathway is catalyzed by one single enzyme, arsenic 3-oxidation state methyltransferase, AS3MT. Now, there is a new mechanism or newer mechanism that was proposed in the lab of Dr. Barry Rosen in Florida International University. It's basically a reductive methylation, but instead of using glutathione, which we know today is not essential actually for methylation of arsenic, this mechanism proposes that cysteines that are found in active center of arsenic methyltransferase keep arsenic in trivalent state through the two methylation steps, and that the pentavalent arsenicals, which are also commonly found in urine, for example, are actually a result of spontaneous oxidation of the trivalent species here. So this is probably the most relevant mechanism so far because it's based on thorough study of the structure of the active enzyme, of the active center of the enzyme. So one thing we know for years actually, but kind of didn't know what to do about it, is the fact that the efficiency of arsenic methylation differs among species, with some species actually not being able to methylate arsenic, like chimpanzee or marmoset monkey. The efficiency of arsenic methylation is generally regarded or evaluated looking at the ratio of dimethyl to monomethyl arsenic, or percentage of methyl arsenic and dimethyl arsenic in urine of exposed individuals. The higher percentage of DMA, the more efficient the methylation is, which, again, we are looking in urine. I have been always cautious about interpretation of this data, but that's the general sentiment here. Now, the truth is that there's a big difference in percentages or proportions of these methylated species in human urine versus some animal urine. And that is particularly true about urine of mice. So in mice, typically, all urinary arsenic, or almost all, is dimethyl arsenic. And it really doesn't depend on dose. I'll show you some results later on. In humans, based on multiple studies, the proportions differ. There is much more or much greater proportion of inorganic and monomethyl arsenic and less of dimethyl arsenic, suggesting that humans may be less efficient in methylating and therefore detoxifying arsenic. Again, based solely on urine, because we don't really have much data on tissue concentration of arsenic from human studies. Unfortunately, after all these years in arsenic toxicology, we don't have data. So one other evidence that human metabolize arsenic less efficiently than mouse comes from this table. This table shows values of arsenic, total arsenic in tissues of man exposed to 3 different concentrations of arsenic in water, assuming 2-liter water consumption per day. And these numbers here are not actually measured numbers. They are based on a PPBK model that was developed at US EPA about 10 years ago by Dr. Al Master's team. What I did when I was applying for a grant a year ago, I filled in this table levels of arsenic, total arsenic we found in mice in our experiment, specifically C57 black 6 mice, exposed to the same levels of arsenic. And one thing you can see clearly from this chart is that levels of total arsenic in human tissues are consistently much higher at the same exposure level than mice tissues, both males and female mice. So this comes from multiple mouse experiments from our laboratory, and our laboratory has this advantage that we can actually measure arsenic in tissues. So this is particularly visible in the liver. Look at these differences here. So that tells you, again, this is consistent with the observation that percentage of dimethyl arsenic in urine is lower in human urine compared with mouse urine, suggesting that there is much more arsenic retained in tissue, which is confirmed or supported by this model in comparison with experimental data. So now you can think that maybe this applies only to this mouse strain, but we have been showing this consistently in other mouse strains. This study was published 2 years ago, in which we actually look at metabolism of arsenic in genetically diverse collaborative cross strains, 12 of these strains, which are very different in terms of the genetic makeup, but all of them obviously express mouse arsenic methyltransferase. They were exposed for 2 weeks to either 0.1 ppm or 50 ppm arsenic. And this is how distribution of arsenicus look in urine: green dimethyl arsenic, orange monomethyl arsenic, blue inorganic arsenic. So you see that what I said before, that most of the arsenic in urine of mice is dimethyl arsenic, is true. It's more than 95% in general. And it's not that different between these strains, although they are quite different by their genetic makeup. Now, the dose really doesn't change much here. 50 ppm still comes back with mostly dimethyl arsenic in urine. What increases is monomethyl arsenic level. So that tells you that neither of these mouse strains are really good model for metabolism of arsenic in humans. And that just stresses the point I'm trying to make, I've been trying to make for a couple of years, that mouse, although we have used mice for a long time, is not the best model. So what we came up with an idea with my colleagues in genetics at UNC, that if we humanize arsenic methyltransferase gene in mice, we may end up with a more human-like pattern of arsenic metabolism. Now, that was an idea, and I need expertise of my colleagues in genetics, specifically Dr. Beverly Kohler's lab, to actually do an experiment or create a mouse strain that would have humanized arsenic methyltransferase gene. Now, what she suggested to do is not to humanize only arsenic methyltransferase, but also the adjacent gene, which there is a typo here, it says BORX7 gene. It's adjacent to arsenic methyltransferase, and some data suggest that there is an overlap between BORX coding region or non-coding region and the 5' UTR region of arsenic methyltransferase. And the same paper suggests that there could be an influence of BORX on the transcription arsenic methyltransferase because of this overlap. So meaning Dr. Kohler, suggested, why don't we replace both genes? And that's what she did. She deleted the 2 gene segment from a mouse genome. She replaced it with human segment. I mean, she would describe this much better than I would do, but she generated first embryonic stem cells that were derived from 129S6 mice. She deleted the mouse genes and expressed the human genes by homologous recombination. She selected the properly expressing stem cells that express human allele, and she first created chimeric 129S6 mice, which offsprings were then mated to create F2 generations. And in our first publication published last year, we used this F2 generation to characterize 2 things. First, expression of arsenic methyltransferase, human and mouse, in heterozygotes of the F2 mice, F2 generations, which carried 1 human and 1 wild-type mouse allele. And we use homozygotes that has both human alleles or both wild-type mouse alleles to compare arsenic metabolism in these mice. So these mice come from the same litter, so the comparison is as perfect as it can get. And the arsenic methyltransferase expression was carried out by digital qPCR in tissue of heterozygotes, meaning you can measure expression of the human allele and mouse allele in one tissue from the same animal. And these are results of the transcript levels of arsenic methyltransferase mouse and the human arsenic methyltransferase in selected tissues of these heterozygotes. In white, it's the mouse gene, and in black is the human gene. You see there are substantial differences in the expression levels in these mouse tissues, and what particularly caught our attention was this difference here. The mouse arsenic methyltransferase was expressed very highly in the liver, not so for human arsenic methyltransferase. And the opposite was found in adrenals, where the human allele was expressed by 4 times higher levels than the mouse allele. And this appears to be consistent with prediction of the arsenic methyltransferase levels in human proteins, in human tissues based on the human protein atlas. So this is a very intriguing finding, because just looking at the transcription level, these are not protein levels, these are just transcript level. It would suggest that mice may be less, these humanized mice might have less capacity, a smaller capacity to metabolize arsenic, at least in the liver, which is thought to be a central organ for metabolism of arsenic. Now, adrenals are really interesting here with this high level of expression, and we were able to show that it's both cortex and adrenal gland that express the human allele of arsenic methyltransferase. Obviously, also BORX7. Now, there was one interesting finding here, which may or may not be related to arsenic toxicology. Human literature suggests that there is a specific splicing variant in neural tissues, including brain of humans. It's a D2D3 truncated splicing variant of arsenic methyltransferase. One single paper that is published suggests that this variant does not methylate arsenic, although the assay they used is kind of obsolete, not very reliable, but it is possible because of the truncation. Now, what's interesting from the human health point of view is that this truncated form in human brain was linked to risk of schizophrenia. Whether or not it has something to do with arsenic exposure, it's too early to tell. But what's interesting, these humanized mice express this human-specific splicing variant in their brain. So it calls for further investigation or using these mice for research of schizophrenia in context or out of context with arsenic exposure, which is something we hopefully will be doing sometimes in the future. So I said, so that was done in heterozygotes. The homozygotes, wild type and humanized, were used to study arsenic metabolism. I need to work time. So we did 2 types of experiments here. First was an acute single dose, oral dose, 20 micrograms of arsenic per kilogram body weight, which is a moderately high dose. And we collected feces and urine metabolic cages for 3 days in 24-hour intervals. The second type of experiment was subchronic exposure. For 4 weeks, mice were exposed to 25 or 400 micrograms of arsenic per liter ad libitum. In both cases, we use sodium arsenite as inorganic arsenic. Now, the 400 ppb data are published in part. I will show you some data from 25 ppb exposure. We collected urine, spot urine samples, and we collected tissues at sacrifice and measured arsenic in both urine, feces in case of acute exposure, and tissues. So for a single oral dose, what you see here is a total arsenic level in urine in 24-hour intervals after dose for a total of 3 days. And same for feces on the right side. What you see here right away in 24 hours is that the humanized mice that are labeled as HSHS here, M stands for male, F stands for female, and the wild type homozygote differ quite a bit in levels of total arsenic. The wild type has much higher level of total arsenic in urine during 24 hours after this single dose. There was little difference in the following 2 24-hour intervals, and totally in 3 days there was much more arsenic, significantly more arsenic excreted in urine of wild type versus humanized mice. So here you go. These mice are not, the humanized mice are not very good in getting rid of the arsenic, and this is % of dose. So you see that even after 3 days, there is a substantial amount of arsenic still remaining in tissue. Only about 30 to less than 40% was excreted, compared with what was 50 and almost 80%. There is also interesting sex difference here. Now in feces, the proportions are opposite here. And the biggest difference were in after 24-hour time interval, where it were the humanized mice, males and females, who excreted more arsenic in feces. And again, that's also true about total arsenic in 72 hours. There's actually very little, if any, data on excretion of arsenic in human feces, and hopefully this model can tell us a little bit about this. So again, this is the ultimate result of this single dose experiment. So this is total arsenic. So obviously we are interested in proportions of arsenic in urine, and that's what this chart shows. First 24 hours, second 24 hours, and finally the third 24 hours. The proportions are inorganic arsenic in white, black is monomethyl arsenic, and gray is dimethyl arsenic. So again, comparison of the humanized males, wild-type males, humanized females, wild-type females. You see a striking difference in proportions of arsenic levels, which changes to some extent as time goes after the dose. Ultimately, what you see here that wild-type mice again excrete almost entirely dimethyl arsenic, and humanized mice have significant proportion of urinary arsenic in the form of inorganic arsenic and monomethyl arsenic, which is true about both males and females, although there may be some sex differences here. In feces, again, the differences appear to be most significant after 24 hours. Just some, yeah, too much animation, right? So here what you see is that in both genotypes, it's actually inorganic arsenic in feces. That is a majority, which is the major metabolite here, but the humanized mice feces contain a lot of monomethyl arsenic, which is barely seen in feces of the wild-type mice. So striking differences again between the humanized and wild-type mice. And again, this is fairly new because I don't think we have any data on excretion of arsenic in feces. So for subchronic exposure to 400 micromolar, 400 microgram arsenic per liter for 4 weeks, first data I'm showing you are total arsenic in spot urine samples during the first week or after first week, second week, third week, and fourth week of the exposure. You see that the most dramatic differences between humanized and wild-type mice are seen during the first week. and they kind of leveled off for the fourth week. But this is total arsenic again. See that in general, the humanized mice do not excrete much of, or less total arsenic than wild-type mice, at least for the first 2 weeks of the exposure. When you look at proportion of arsenicals after 1 week and after 4 weeks, you see proportions are similar to what we see after single dose. Seeing dimethyl arsenic in urine of wild-type mice with significant proportions of inorganic and monomethyl arsenic in urine of the humanized mice. So this is consistent with single dose and tells us again that humanized mice are not efficient methylators, producing much less dimethyl arsenic. Now, how about tissues here? Okay, so before I go to tissues. So we have run recently experiments in which we also exposed mouse to 25 ppb arsenic. And the reason we did it because one of the reviewers of our grant suggested that what we see here, the less efficient methylation of arsenic, may have something to do with the dose. 400 ppb being very high dose, also not much for mice. So maybe we are saturating the system. That's why we are seeing so much inorganic and monomethyl arsenic. And the reviewer suggested, why don't we do it in low levels, which would be more relevant to human exposure? So we did. And this is what you see when you compare urines of wild type and humanized mice, total arsenic in urines after 425 ppb. You see that the same differences we saw for 400 ppb are even more pronounced after 4-week exposure to 25 ppb. It's again the humanized mice that excrete less arsenic than the wild-type mice. And the proportions are consistent in both doses with humanized mice urine containing a lot of inorganic and methyl arsenic as compared to mostly dimethyl arsenic in urine of wild-type mice. So it means that dose is not really a decisive factor here. The mechanism of methylation is simply inefficient regardless of dose, although other factors, not just methylation, may play a role here. So what about tissues? When you look, and that this is a critical information because looking at the urine doesn't tell you whole story, right? Which is certainly true about the human studies. Now here we can actually analyze arsenic in tissues. This is how total arsenic in the liver of mice exposed to 425 ppb arsenic look like. Much higher levels in the humanized mice tissues with female livers actually containing higher levels of total arsenic than in male liver. And it's pretty consistent for the doses. Again, actually larger differences between genotypes are seen at lower dose, not at the higher dose. The proportions of arsenicals in liver looks like this. Again, for big differences between the genotypes with a lot of inorganic arsenic contained in the liver of humanized mice, actually for both doses, with prevalent dimethyl arsenic in livers of the wild-type mice, especially at high dose as compared to low dose. So saturation is not really an issue here in any way. Something else is playing a role here, and the something else is the human enzyme. So this is how kidney looks, and you will see the pattern repeats in other tissues. Again, it's the kidneys of humanized mice that contain much more total arsenic, and the proportions actually are interesting for kidney because the proportions favor monomethyl arsenic in kidneys of the humanized mice. Now, I always say that if you see arsenic within a tissue, within the cell, it's going to be trivalent arsenic because of the reducing environment. We can actually measure valency of arsenic in these tissues. We didn't do it in this experiment. But if that's true, then monomethyl arsenic in trivalent state is the most toxic arsenic metabolite. And I think very few labs are looking at arsenic effects in the kidney. So this may be a great model to do just that because of this accumulation of monomethyl arsenic. Now here is a similar picture in adrenals. We were curious about adrenals because of the differences in expression levels of arsenic methyltransferase human versus mice. And these are slightly different here. We actually, at low dose, we don't see significant differences in total arsenic between the genotypes. We see these differences only at higher dose, and again, consistent with the other tissues. The proportions of our arsenicals also are not that different in adrenals of wild types and humanized mice at low dose. It's a lot of monometal arsenic being present again. But at higher dose, it again shows more efficient methylation for the wild type mice as compared to humanized mice. I have some other tissues, but I didn't want to bore you because the pattern is really consistent with what I have already shown you. So what we did at the end, we actually fitted the concentration of arsenicals in liver and kidneys of the humanized and wild type mice, males and females, into the table along with the predicted levels of total arsenic generated by PPBK model at US EPA by Dr. Miroslav. And at 400 ppb, these are predicted levels of total arsenic in human liver and kidney. They compared quite well with what we are finding in the liver and kidneys of humanized mice, and specifically males. Females tend to actually have much higher levels of total arsenic with humanization of arsenic methyltransferase. Now you see that the wild type mice have much lower levels of arsenic in the liver. With 25 ppb, the story is somewhat intriguing because it shows these levels, these are micrograms per kilo, by the way, of total arsenic in human tissues, but higher concentrations in humanized mice tissue, and again, very low in wild type. Now, there is one issue that we need to consider, though. We exposed these mice to 25 ppb arsenic in drinking water. We fed them semi-purified diet. Now, if you are following literature on arsenic, you know that diet can actually contain a lot of arsenic, especially not the purified one, up to 400 ppb arsenic. In some diets, it's actually mostly inorganic arsenic, which is, again, a factor you need to consider if you are studying arsenic at low levels. So here we had semi-purified diet, which consistently showed levels of arsenic up to 40 to 50 ppb. So in addition to 25 ppb in water, these mice were also exposed arsenic in diet, which may explain that these numbers are higher than those predicted for humans. It still suggests that the humanized mice much better reflect or model the human metabolism of arsenic. So for this part of the study, we can conclude that we have generated or created, my coworker, Dr. Kohler, doesn't like the word created, so why not created, right? Humanized 129S6 mice, which express human arsenic methyltransferase and BORC7, in which metabolism of arsenic resembled those described in humans, at least based on urine, because we don't have tissue values for arsenic. The pattern of arsenic metabolism suggests that the humanized mice are much less efficient in metabolizing detoxifying arsenic than wild-type mice, and therefore we expect them to be more sensitive to arsenic exposure because they accumulate much more inorganic and monometal arsenic detoxic forms in the tissues. So we can further modify these mice because even I don't believe that arsenic methyltransferase is the only important player in arsenic metabolism. So we can either use CRISPR to introduce single nucleotide polymorphism in the arsenic methyltransferase gene. We can humanize another gene if needed. My tip would be to look at transporters of arsenicals, which may again explain some of the differences between mice and humans. So that all could be done, and some of it will be done. I have very little data on phenotype apart from arsenic metabolism, but I thought I would share with you some of the preliminary data, which still have to be confirmed. Please take this as very preliminary. By phenotype we are interested in is phenotype related to diabetes. So we measure insulin, we measure blood glucose, fasting blood glucose specifically. We do some other tests. In this study, being only 4 weeks along, we didn't expect actually much going on, but we did see statistically significant difference in fasting blood glucose levels, but between males, wild type and humanized mice, not in females, with fasting blood glucose level being higher in the humanized mice, suggesting diabetogenic effects of arsenic. There is a similar difference in 25 ppb, but not statistically significant. But if you look at the average value of fasting blood glucose, it's about 135 here, and it's about 165 here. So it takes probably additional arsenic exposure to see significant effects. Now, one additional phenotypic characteristics we are interested in are levels of adrenal hormones. Again, this was a quick experiment after 4-week exposure only. But it appears that there is a statistically significant difference in fasting. This is fasting plasma between the humanized and wild type mice. It says, I'm sorry, this should say 400 only, not 25 micrograms per liter. This is only for 400 ppb exposure mice. Again, the difference is in males, not in females. We keep seeing these intriguing sex-related differences throughout the humanized mouse studies. Something probably to pay attention to, because we don't have much data from human population studies. So what we are planning to do in a near future, having R01 grant now to do that, first thing we want to do is to transfer the mice from 129S6 phenotype to black 6 phenotype. The reason is that if you look at literature for 129S6s, they are not really responsive much to pro-obesity or pro-diabetogenic effects. Even on high-fat diet, they don't really become much obese. They don't become diabetic. And diabetes is an endpoint we have been studying in our lab. So although we saw this shift in fasting blood glucose, we think that using black 6 background will provide much better model than humanized, right? So we are almost done with, so it's transferring these mice to black 6 background, hopefully having almost pure congenic mice ready in a couple of months. We obviously want to use these mice in our diabetes-related studies, looking at the actual effects of arsenic exposure for a prolonged time, not just for 4 weeks. Now, we are really intrigued with these high levels of expression of human arsenic methyltransferase in adrenals. The very preliminary hypothesis is that arsenic methyltransferase may actually play some role in adrenal function. So we are, our project includes looking at the production of steroid hormones and not just adrenals. So we are looking at HPA axis in a broader approach, but also including not just chemical tests, but also including behavioral tests. We are planning to create mice that would express so-called risk human arsenic methyltransferase haplotype. And the plan is to introduce in the existing humanized mouse arsenic methyltransferase gene SNPs that have been linked to either inefficient methylation of arsenic or a risk of DZ in arsenic-exposed populations. So based on population studies strictly, hopefully we are in particular interested in the methionine 287 threonine variant, which has been linked to both inefficient metabolism and risk of some arsenic-induced diseases. And finally, we are on the track to generate an additional set of interesting humanized mouse models in collaboration with Dr. Manuel de Viana, who is chief of genetics at UNC, with huge experience in genetically modified mice and with collaborative cross mice. Sorry about that. We are on track to generate several, actually many, collaborative cross mouse lines that will be expressing human arsenic methyltransferase. And I have a couple of slides that I want to show you because I found it very exciting. So those of you who are not familiar with collaborative cross was created by mating 8 donor mice or founder mice. These mice are listed here. There are some very obvious mouse strains, including C57 black cyst or NOD mice, or this is actually wild mouse coming from Prague, my hometown. So that's interesting. And those are being or have been mating, cross-mating using this funnel scheme, which I'm not going to details to explain it because I'm not able to. But the point is that at the end, about 60 mouse strains were generated that were eventually inbred that have completely different genetic makeup. The goal obviously is to have mice that would resemble to some extent human population. So you can actually study a role of genetic diversity on various outcomes using mice from various strains. So it's similar to DO mice, diversity outbred mice, if you are familiar with that, but these are inbred, so you can actually have a population from several strains with exactly same genotype, while differences are between the genotypes. So this is just an example of how the genetic makeup look in 4 of these strains. We actually selected these strains for humanization on arsenic methyltransferase and BORC7 gene. So these, just let me go here. So you see these colors here, which stand for each of the founder mouse strains. Here you see combination of these colors in chromosomes of 4 CC mice. This is how they are marked: CCO28, 57, 78, and 61. You see that they are very different in how the genes from founder strains are combined in their genome. Now, Dr. Manuel de Viena is using an expedited way of generation of congenic mouse from these collaborative cross strains, instead of just doing 10 times mating and generating offspring in 10 or more generation. He is mating a collaborative cross mice with the 129S6 humanized mice, and in the first generation and any following generation, he's using mini-MAGA genotyping to actually identify an offspring that has most enriched background and with least background of the original 129S genotype. So it is shown here on these charts for 5 generations during the crossing. The red color in chromosomes stands for the original 129S6 genome types, and you see that after 5 generations, he ends up with congenic 99.3% congenic mice on the selected CC background. So this is almost done now. We now have these 4 mouse strains that are congenic by more than 90%, and he is generating additional mice. So what we are trying to generate here are not just humanized mice on arsenic methyltransferase gene, but also mice that have very different genetic background, the way you see it in human populations. So this will allow us to study not only the role of the human arsenic methyltransferase in mice, but also doing this study in context with a diverse genetic background. So this is a very exciting project which promises a number of new models for arsenic toxicology. And that's really all I have for you today. And I already mentioned Dr. De Viena as a leading person on humanization of CC mice, but I want to thank in particular Dr. Bev Kollerslap and her co-worker John Snoward. Unfortunately, I wasn't able to get a better picture of Bev. She hates to be photographed. So these guys are really guilty of creating this mouse strain. I was just testing the metabolism in these mice. Dr. El Masri and Dave Thomas were helping us with data from the PPBK model, comparing the human model data with our actual measures on of arsenic metabolites in mouse strains. And my lab, now this is kind of outdated picture, but these are my current coworkers who are heavily involved in this work. Christelle Duet, I should say Dr. Christelle Duet is my lab manager, and she takes care of everything in the lab, including all mouse studies, and also involved in analysis of arsenic in biological samples. Madison Miller is my master's student who is currently running studies that would evaluate the phenotypic changes in humanized mice exposed to arsenic. Pete Cable is a recent hire who is working on our ICP-MS instrument for speciation of arsenic, and Qing Shi is a laboratory technician who is taking care of everything in the lab relating to instrumental maintenance and odors. And that's all I have for you. And there is time for questions, I believe. Oh, I should have thanked to grants from CIEHS. I don't think we could do it without those.
How about the answer question from the audience here first?
Yeah.
And can help you to find the question from online. Okay.
Outstanding. Something struck me is that the metabolism of arsenic seems to be greater in the wild type like or no, I'm sorry, the other way around.
The other way around. Yes. It doesn't suggest saturation, does it? Which, you know, we still know very little about how arsenic exposure affects the expression of arsenic transferase, which could be one explanation, right? There seems to be a shift in the monomethyl arsenic levels, including those data in collaborative cross, those 12 strains I show you. There is really more monomethyl arsenic when you expose mice to 50, wild type mice to 50 ppm as compared to 0.1 ppm. But these mice, 50 ppm is not really any risk exposure to these mice. They can live on it for months with no signs of losing weight. One sign, if you put them on high-fat diet, they actually become leaner, I guess, due to arsenic exposure. But I don't have explanation for that. For this, you know, difference between low and high dose metabolism, I think it may have something to do with expression of arsenic methyltransferase or other genes, or that are involved in arsenic metabolism. And I was suggesting maybe arsenic transporters is something we should be looking at closer than we have so far. But I agree, this is surprising. It definitely doesn't fit to what has been published in human populations, where a couple of studies suggesting that high exposures are associated with a lower percentage of DMA in urine, though. So, but those, if you look at those data, we're talking about a couple of percent point difference. It's not really striking. It's significant because you have hundreds of people. We have, a dozen of mouse here. So yeah, I don't have better explanation.
Very nice, thank you. When it came to the expression levels of mouse versus human transfer, you showed that there was a striking difference in liver and human. Whereas the human expression transcript was low in liver but higher in renal. But when you expose these mice to arsenic, actually the tissue levels of arsenic was higher in both cases. So I'm wondering whether metabolism of arsenic has anything to do with tissue accumulation in these mice.
Well, it certainly does in a global way, right? But what the arsenic retained in mouse tissue is not necessarily product of methylation in that tissue, right? It really is about the distribution of arsenic species between the tissues. We hypothesize that most of the arsenic given by oral exposure, right, is methylated in the liver, assuming that portal circulation would take arsenic first to the liver, where most of it would be methylated, and both remaining inorganic arsenic and methylated arsenic will be distributed throughout the tissues. We really don't have good data on capacity of other tissues to methylate arsenic. I was just talking to colleagues from Dr. State's lab that we found surprising methylation capacity in adrenals. In pancreatic islets, surprisingly high. So what I'm trying to say is it looks paradoxical, but if you think about that, the methylated species in adrenals are not necessarily originating in adrenals. And if they are, they may be excreted from adrenals much more efficiently than, for example, from the liver. So there is a lot of dynamics going on between the tissues, which really don't let us tell what tissue is the better methylator than other. We are actually running currently, one thing we want to do is to compare methylation capacity of adrenals of wild-type mice and humanized mice in vitro, just using tissue homogenates. It doesn't go very easy so far, to tell you the truth. We can follow methylation in adrenal homogenate, but we don't think our system is optimized at this point. So that would tell us directly, how much, what is the actual methylation capacity. We think we will be able to do it at some point. We can probably do it in the liver even better using primary hepatocytes from wild type and humanized mice, which is also in plans in next few months. But yes, this is a very good question. What's produced in the tissue and what's retained in the tissue and where it comes from.
Hi. Oh. May I ask a question, or is someone asking? Oh, okay.
Everyone in the room has asked a question. Then we'll switch to online. And I'll call on people's names that are raised if they raise their hand.
I was curious, in your subchronic exposure studies, it looked like metabolism of arsenic was changing over the duration of exposure. Are the transcript levels of the two alleles changing?
Yeah, that goes on with Chris's question. It does suggest that the system is modified by exposure, and it becomes more efficient. If you compare with first week of exposure with 4 weeks of exposure, you actually see more dimethyl arsenic percent-wise in urine. So again, as you suggest, arsenic exposure is known to affect gene transcription for many genes in many organs or cells. Interestingly, there is very little data on transcription of arsenic methyltransferase or expression of arsenic methyltransferase. We have shown in one cell type a long time ago that arsenic exposure may increase transcript of arsenic methyltransferase. We didn't see it on other cell types, but those are all mouse cells. So yeah, it's a logical explanation that there is an activation of transcriptional processes due to arsenic exposure, which may or may not include arsenic methyltransferase. And that's something we can answer. The transcripts I showed you on these presentations were in mice that are not exposed to arsenic. I should probably correct myself because there is arsenic in mouse diet. Okay, and one big problem we are having right now, we are keeping our humanized mouse colony on regular diet, grain-based diet, simply because it's just too expensive to buy pre-purified diet to generate large colonies of humanized mice. So these mice are exposed generation by generation to arsenic in diet, which in a regular grain-based diet could be easily 100 ppb in organic arsenic. And there is no way to avoid that. So those mice may be modified already before we actually take them for experiment. And something to keep in mind when you do these experiments, especially for low-level exposures in drinking water, that there is exposure before you even start the experiment. And you just need to make sure you control for every aspect you can, even if you cannot change it. I probably didn't answer your question directly.
One more question you have, right?
I was going to ask a question which is very similar to what Dr. Watson asked, but instead of the duration, my question was, is the sex have an effect sex and arsenic have an effect on how the human and the mouse are being expressed in the tissue?
When you're asking about expression, I have no idea. When you are asking about, or if you, I can answer a different question, if it has effect on capacity to metabolize arsenic, which may or may not have something to do with expression. Human studies do suggest, some of them, that women may be more efficient in methylating arsenic just based on this percentage of DMA in urine, which I think is a very limited evidence, but it kind of matches to what we see here between humanized and wild-type mice. Pregnant women seem to be even better methylators based on Dr. Wachter studies. What is behind that? I don't know, and I don't think anybody knows. And correct me if I'm wrong. I think.
Okay, and we have many hands raised online. Dr. Frank Gross, if you would like to go first.
Thank you. You mentioned that you have data and you didn't want to bore us, quote unquote, with data on the other organs where arsenic would be accumulating. But since it's being excreted in both urine and feces, have you looked at what happens downstream? So what does it do in the bladder, the ureter, urethra? What does it do in the colon as it passes through?
By what does it do? You mean the effect on functions or cell properties or metabolite retention?
Metabolite retention. I mean, that's what you've been looking at. But
Okay.
if you have something to share about the effects, by all means.
Well, you should realize that this mouse strain was created just in the last 12 months. And the only thing we could do so far, not having grant, which came just this month, was to look at metabolism of arsenic and just to document how different it is from wild-type mice. But your question is a logical question to ask and logical question to follow, and that's what we are proposing to do in the next steps. But we will be looking specifically on effects and mechanisms that are related to diabetes. That's what we have done before in wild-type mice. We want to see what we can observe in humanized mice, meaning we will be looking at beta cell function and insulin resistance related signaling. We are offering these mice to whoever wants to study any other effects in any other tissues. Bladder, you mentioned bladder cells. This bladder is one of the targets. We have shown as the only lab in the world that arsenicals are retained in urinary bladder cells, or better, urothelial cells that are exfoliated and excreted in urine. We published that paper on a long time ago. So I think in human it's difficult to link metabolism in tissues to the effects simply because very few labs can analyze tissue arsenicals, and very few labs have access to these tissues. But these mice hopefully will let us do that on the background of human-like metabolism of arsenic. So I'm sorry I cannot answer your question directly, simply because it's just too early to answer it.
Thank you.
And then next is Dr. Caldwell.
Hi. So I think my question was answered. Oh. So I think my question was answered earlier about why we were seeing that females had, they were better metabolizers of arsenic, yet they had more storage in their tissues. And I think that you had speculated and answered that it's unknown as to why that happens and why it occurs. But are you looking at any other hormones when you're going to do your panel for casting? Which ones do you think that you might look at?
We are not looking at hormones right now, but the plan is to look at hormones, both transcription and circulation, specifically related to adrenals and HPA axis. So yeah, another logical question. Again, I need to remind you again that this mouse strain was just created, and it will take probably years.
Which is great.
Yeah, I know. So the plan is to characterize these mouse strains, looking at black 6 mice actually, rather than humanized black 6 mice. in depth as much as we can, including doing transcriptional profiles in key tissues that key for us, okay? Key tissues may mean something else for you. And again, there is a lot of work ahead using these mice, assuming that we can generate enough mice to satisfy all the requests we are receiving right now.
Yeah, and then I had one last question. I think I might have missed it. How old were the mice when you had exposed them?
Yeah, that's a good question, isn't it? So it's in the paper published in EHP, and I, being old man, don't remember. They were not old mice, but they were not wean mice, meaning they were a couple of months old before we exposed them. Sorry, I really don't remember. But if you look up our paper in EHP, Environmental Health Perspective, it has all the details because we had to provide all the details. Otherwise, whoever published in EHP know what I'm talking about. It's a process.
Wonderful. Thank you so much and great talk.
Thank you.
All right, and then next is Dr. Jamie Young.
Hi, Mirek. Can you hear me?
Yes.
Okay, great. Yeah, fantastic talk. I have a more general question about humanized mice because I'm thinking about working with some in the near future, and I wonder about like off-target effects when you have like a human gene interacting with the other mouse genes. So it's great that we see the humanized, transferase or this receptor, and that we might see more of what we would see in an epidemiology setting when we use these models. But as we like use these mice as they grow older and as they go through different translations, do you think that there's any chance for like an odd or off interaction between the mouse genes and the human genes that may give us some, outcomes that we're not expecting?
That's
or
an
am I
interesting.
really off on that?
I hear you. I do understand your question. I just don't feel have enough expertise to answer it. Frankly, this would be a question for my collaborators in genetics. But yes, this is a good question. The fact that we are expressing not only human arsenic methyltransferase, but also BORG7 may be important because BORG7 knockout is lethal at embryonic stage. And assuming that these genes have larger regulatory functions, especially BORGs, could mean that introducing human gene to mouse genome would have some consequences, as you suggested, for regulation, transcription of mouse genes, which ultimately can affect the endpoints we are interested in, right? Now, the problem I see is technical here. We will never know. And the reason is that what I already said, all these mice have been exposed to arsenic through diet as they are being bred. So when you compare, let's say, transcriptional profiles between the humanized mice and the wild-type mice, looking at specific tissues or cells, and you will see differences, I'm pretty sure about that, right? Even unexposed, okay? But unexposed, I mean conditionally because they are exposed to arsenic in the diet. And when you see the difference in mice that you are not exposing through drinking water, the difference could be either because the introduction of human genes affect transcription and regulation of mouse genes, or because they have arsenic in diet and they respond differently to arsenic in diet. And that has been, major conundrum we have been dealing with in our lab, because people now keep publishing these studies where they expose mice to 10 ppb arsenic in drinking water. And okay, I'm not criticizing these studies because that's what we want to do. But if you use regular grain diet, you have 100 ppb of arsenic in your diet. So how can you know that it's the arsenic in water? Or anyway, it's arsenic in diet is a big deal for generally for mouse studies, but for humanized mouse study even more because they will accumulate this arsenic to a greater extent. And I don't have data here because all the mice I showed you were exposed to arsenic, but I have data in mice that are not exposed to arsenic in drinking water. They have a lot of arsenic in tissues, and humanized mice have more arsenic in tissues than wild-type mice before we expose them to arsenic in drinking water. So ideal experiment would be to have super clean, arsenic-free diet on which we can breed these mice. because again, all these exposures in generations could affect epigenetic mechanisms and transcriptional profiles in next generation, and what we are dealing with is something that has already been changed by arsenic. I don't know if I answered your question, but
it was more of a food for thought type of thing that came to mind. I think the idea of the humanized mouse models is important, and I think it, giving the relevance to what we see in the epidemiology, they are important. It's just something to consider as we move forward using these type of models.
Well, one thing to consider in epidemiology, if I can say or give you some thought. So if BORG7 is really affecting transcription profiles of arsenic methyltransferase, then maybe looking at SNPs in BORG7 could be an important thing to do in epidemiology when you are evaluating arsenic metabolism alone or arsenic metabolism as a factor affecting risk of human disease. I know there were genome-wide studies, and BORGs. I don't think BORGs came up as a gene that would be involved in differences in arsenic metabolites in urine, but then it's urine.